This video is about the microorganism Toxoplasmosis gondii. So first off, right off the bat, I already said it, this organism is a parasite. And that means that it's a eukaryotic organism. So what does that mean? Well, see, it means it has a nucleus. See? It also has a whole bunch of other organelles like endoplasmic reticulum, Golgi body, mitochondria, parasites. They're just like us, except not. Because while it has all of these things, it's also a protozoan. So what does that mean? Well, protozoans are one-celled organisms or single-celled organisms, which we aren't. And it's also capable of anaerobic metabolism, which makes it a facultative anaerobe. T. gondii is a certain kind of parasite, and it belongs to the subclass Coccidia. This really just tells you about its taxonomy, but it also would tell you, if you knew the taxonomy of parasites really well, that it's a microscopic, spore-forming, single-celled intracellular parasite that infects the intestinal tract of animals. There's really only one strain of T. gondii with very little variation, and there's only one species of T. gondii, so it's pretty easy to track. T. gondii can be found in all sorts of animals, birds, cows, humans, to name a few. And as with all parasitic infections, its life cycle and how it is transmitted to us as a pathogen are inextricably linked. So here's the life cycle of the organism T. gondii. Do not memorize it. I'm just showing it to you to orient you. There's only one parasite whose life cycle I will ask you to memorize, and it's Plasmodium, the organism that causes malaria, not this one. But there are some things that we kind of need to point out about this particular life cycle. So when we're talking about parasitic infections, it doesn't matter if we're talking about T. gondii or P. falciparum. There are three questions that you need to ask, and you need to know the answers to these as they relate to the life cycle of the organism. This doesn't mean you need to memorize every life cycle, it just means that you need to be able to answer these questions. The first is, what is the infective form? What form is the parasite in when we encounter it? Is it an egg? Is it an amoeba? Is it a worm? How are we seeing it? The second one is, what is the disease-causing form? So parasites tend to develop in hosts. So we're a host, birds are hosts, cows are hosts, kind of depending on the organism. So we might encounter it in one form, in the infective form, but then it might take on a different conformation in its developmental cycle that is what causes disease. So again, are we talking about an egg, a cyst, maybe a worm? And then the last one is, what is the diagnostic form? So just like with when we see it versus when it causes disease, what tests have we developed at what stage in the parasitic life cycle are we able to detect? Are we able to detect the eggs, the cysts they form, the worms? I've got kind of a thing about worms. I'm a little freaked out if you couldn't tell. But anyway, you need to answer these questions. Now let's get him off the screen because he's super scary. So with those questions in mind, how do we answer them about T. gondii? So we're really talking about three things with T. gondii life cycle. We're talking about the oocyst, down here an immature oocyst or a mature oocyst. These are basically a cyst that contains the zygote or eggs of a parasite. We're also talking about these things, tachyzoites. Tachyzoites are the fast replicating forms of T. gondii. And as the organism continues to develop, they'll become bradyzoites. Bradyzoites are basically the slow replicating forms of the T. gondii parasite. So we need to keep these three structures in mind: the oocyst, the tachyzoite, and the bradyzoite. There are three major ways that T. gondii is transmitted: foodborne transmission, zoonotic or animal to human transmission, and congenital or vertical transmission, which is mother to child. Foodborne transmission is the most common, and it's caused by ingestion of undercooked meat containing the encysted bradyzoites. However, both humans and livestock, like cows and pigs, can become infected by accidental ingestion of soil that contains T. gondii oocysts. As a result of poorly washed fruit and veggies or by drinking water contaminated with oocysts, that's typically how we get infected from contamination, more so than our livestock friends. So, how do the oocysts get in the soil to infect our livestock or us anyway? Well, the answer, dear friends, is unfortunately man's not so best friend, the cat. These cats are achingly adorable, sweet, little supervillains full of pure evil. They are trying to kill our young with their poop, literally, because these oocysts are shed from feces, and not just any feces, they are spread from cat feces. T. gondii is specifically spread by the feces of infected cats into the environment or even into the litter box. This is because the cat is an essential reservoir of T. gondii. An essential reservoir is an organism that the parasite needs to spend time in order to complete its development. So when it's an essential reservoir, this means that the T. gondii literally requires time in the intestinal tract of felines in order to develop. Without the cat, the parasite doesn't develop. T. gondii develops in the intestinal cells of the felines and is spread to the tissues via bloodstreams. The organism passes out of the cat and the feces, where they mature into infective oocysts. So basically, the organism spends time in the intestine, and then the oocysts come out in the feces, and then they continue to mature in the soil before they are picked up by domestic animals, or us. This whole process takes about 3 to 4 days, and then they are transmitted to us via the fecal-oral route. So based on that information, the infective form of T. gondii is the tachyzoite or the oocyst. The disease-causing form is either the bradiozoite or the tachyozoite. In the bradiozoite, it's actually the cysts that are causing the disease. And the diagnostic form is by visualization of either the tachyzoites or the oocyst. So I've alluded a couple of times that T. gondii causes disease. However, that's not technically true. I mean, it does cause disease, but most T. gondii infections are benign and asymptomatic in the immunocompetent host. However, in the immunocompromised host, then we can have some pretty severe illnesses. Symptoms occur as the parasite moves from blood to tissues where it becomes an intracellular pathogen. So when we're talking about it, we're actually talking about two different types of disease. There's acute T. gondii infection or disease, and then there's chronic T. gondii disease. So acute T. gondii disease presents with chills, fever, headache, myalgia, lymphadenitis, and fatigue, kind of non-specific symptoms. Chronic T infection or toxo infection actually presents with all of the same symptoms that you had in acute, but then add in rash or evidence of hepatitis, encephalomyelitis, myocarditis, and in some cases chorioretinitis appears, which might lead to blindness. As I mentioned earlier, the third form of transmission is actually mother to child, congenital transmission, and that results in congenital toxo. Congenital toxoplasmosis is the result of transplacental infection, and it can occur in pregnancy, either from the infection of acquired from meat and meat juices or contact with feces. So when you get exposed to toxoplasmosis during pregnancy, it kind of depends what trimester you're in as to what the readout will be. If it's in the first trimester, you're looking at miscarriage, stillbirth, or potentially severe disease in the child. Second and third trimester transmission has a whole host of symptoms, epilepsy, encephalitis, and you can read the rest here. The most profound of which that we see most commonly is chorioretinitis leading to blindness. This is pretty common in T. gondii infections, which thankfully are not very common at all. Infants may be asymptomatic at birth, only to develop disease months to sometimes years later. So you want to kind of be aware of this infection later on in life if you're seeing some of these symptoms like chorioretinitis and blindness. Most often, children develop the chorioretinitis with or without the blindness or other neurologic problems. You can have chorioretinitis in the absence of blindness or other neurological problems. As I mentioned earlier, in the immunocompetent host, toxoplasmosis really isn't a big deal. It's asymptomatic, maybe a little flu-like feeling, but for the most part, you're going to be just fine and you're going to get over it. In fact, most of us are exposed to toxoplasmosis at some point in our lives. However, in the immunocompromised patient, it's an entirely different story. And this can include a wide variety of patients. So just as we talked about in the listeria video, anytime we're talking about an immunocompromised patient, we're not just talking about patients with HIV and AIDS. We're also talking about patients who are older than 65 years of age, who may have a weakened immune system. We're talking about patients with cancer who may be undergoing chemotherapy, and we're also talking about patients who have received solid organ transplants or bone marrow transplants. All of these patients, for a variety of reasons, have a suppressed cell-mediated immune response. And remember, just like Listeria monocytogenes, even though Toxoplasmosis gondii is a parasite, it's actually an intracellular parasite. So once again, we're relying on our cell-mediated immunity to take care of us. And these patients all represent populations of patients who may have a weakened cell-mediated immune response. The same goes for pregnant women and their children and the fetus, because they have not had time to develop the cell-mediated immune response necessary to fight off a T. gondii infection, which is why they are susceptible to all of those neonatal toxo symptoms. So in the immunocompromised host in older or immunocompromised patients, we're more likely to see what's called reactivation of latent toxoplasmosis. So remember, toxo is pretty ubiquitous. It's found in the soil as a result of, feces in the environment. So most of us have been exposed to it. Some of us have been infected, but our immune response is keeping it in check. When we're dealing with an immunocompromised patient, the immune response might not keep it in check any longer. And at that point, you start seeing reactivated toxo. And the presenting symptoms of reactivated toxo tend to be neurologic. Most frequently, diffuse encephalopathy, meningoencephalitis, or cerebral mass lesions are seen. Reactivation of cerebral toxoplasmosis has emerged as a major cause of encephalitis in patients with AIDS. Although disease is typically associated with AIDS patients, we can see this in other patients who are immunocompromised, who I've recently spoken. Diagnosing T. gondii is actually a little tricky. So the first thing to know is that unlike bacterial infections, you are not going to be able to culture this organism. Culture is non-diagnostic. And the reason is that, remember, this parasite actually requires a lot of different cells to take on its various developmental life forms. So you're not going to be able to just take a sample of this patient's blood and be able to determine on, some form of agar cells if they have T. gondii. So you're going to have to get a little more creative than that. So the first thing people might choose to do is serologic testing. Now, remember, I said that almost all of us have been exposed to T. gondii at some points in our lives. So if we've been exposed to it, chances are we already have antibodies for it. So how can we tell the difference between just, hey, I was exposed to T. gondii as a kid and I have antibody titer versus I have an active ongoing infection? Well, the answer is to look for increasing antibody titers. If the titer is going up at two different time points of specimen collection, and specifically if they are of the IgG or IgE isotype, that's a good indicator that you might have an acute active infection. Presence of IgM antibody, though, that's not diagnostic, especially in pregnant women. IgM antibody for T. gondii is actually kind of a false positive in pregnant women, so you can't use that as a diagnostic marker. There is a specialized serologic profile, the T. gondii serologic profile, that is used by specialized reference laboratories. The TSP tests for antibodies to determine if infection is consistent with recent acquisition or if it's consistent with prior exposure. So this would be another way of identifying whether or not this is just something that you encountered at some point in your life or if you have an active infection going on. The test actually identifies specific subtypes of IgG, IgM, IgA, and IgE antibodies and their reactivity to the strain of T. gondii that causes disease. There are diagnostic measures for the encephalitic and congenital forms of toxoplasmosis. So diagnosis of toxoplasmosis encephalitis usually involves a CT or an MRI with microscopy and either serology or PCR to identify the specific parasite as T. gondii. Congenital or prenatal diagnosis of T. gondii is usually revolving around ultrasonography and amniocentesis, followed by PCR. Serology is really not useful as the maternal IgG might be present. The last thing you could potentially do is biopsy. Biopsy of specimens from lymph nodes, brain, myocardium, CSF, amniotic fluid, or bronchoalveolar lavage can be directly examined for the tachyzoites and cysts. Alternatively, there is a fluorescent antibody stain that can facilitate the direct detection of the organism in the tissue. So treatment, prevention, and control of T. gondii is handled in a couple of ways. So first off, for your immunocompetent patient, there's no disease, so you don't have to worry about them. There's no intervention. For the HIV, AIDS, and immunocompromised patients, it's a little bit more difficult. You're going to use high-dose pyrimethamine, sulfadiazine for 4 to 6 weeks, followed by a low-dose treatment indefinitely for as long as they are immunocompromised. If toxicity develops, you can switch to clindamycin plus pyrimethamine. Trimethylsulfa is an alternative for treatment of disseminated or CNS toxoplasmosis. For the congenital transmission, during the first trimester, there's not really much we can do due to the teratogenicity of the pyrimethamine. So really you're looking at preventative measures. So for prevention, you're going to avoid consumption or handling of raw and undercooked meat or vegetables, and frankly, avoid cat feces. Contaminated litter boxes are of a special concern for pregnant women, as a first-time infection during pregnancy, as we said, can result in transmission of the parasite to the fetus. That's all I have on T. gondii, so check out the next video.